Hello. In this chapter, we're going to learn about viruses, viroids, and prions. First part is about how to differentiate viruses from bacteria. We're going to describe the characteristics of the viruses, how to classify the viruses, how to name them, how to grow them in the lab, how the viruses are identified, and how they do multiply, and list the steps of the multiplication of viruses. So in this table, we're looking at the differences between bacteria and viruses. Remember, bacteria are prokaryotic cells and viruses are non-living organisms. So bacteria, they generally are not intracellular parasites. Some of them, they can be like chlamydias, rickettsias, but for the most part, bacteria are not intracellular parasites. Viruses are always intracellular parasites, so they need to have cells in order to live. Bacteria, they do have plasma membrane for most of them, and for viruses, the viruses only have a plasma membrane if they're enveloped, and the plasma membrane derives from the host cell. Bacteria divides by binary fission, while the viruses, they don't. And bacteria has the two nucleic acids at the same time, DNA and RNA, while viruses, they only can have DNA or RNA, but not both at the same time. Bacteria, they can, some of them generate ATP, but while some others don't. And viruses, they cannot generate ATP, so they depend on the host cell to have energy. And then bacteria, they have ribosomes, so they can make proteins. Viruses, they don't have ribosomes, so they don't depend on the ribosomes from the host cell to produce the proteins. In terms of sensitivity to drugs, so bacteria are sensible to antibiotics, while viruses are sensible to antiviral drugs and also to molecules that we know as, or drugs that we can call interferons or interferons. So what are the characteristics of the viruses in general? So again, they are obligatory intracellular parasites, so they are non-living pathogens, and they contain either DNA or RNA, but not both. And this nucleic acid can be double-stranded, single-stranded, linear, or circular, or it can be segmented or non-segmented. Viruses, they don't have ribosomes, they don't have mitochondria, and around their nucleic acid, they have a protein coat that we know as capsid that protects the nucleic acid. And some viruses, they contain an envelope that is derived from the host cell. Now, some of these viruses, they do also have spikes, and these spikes are protrusions made out of glycoproteins, either from the from the capsid or from the envelope, and these specific glycoproteins are viral attachment proteins that helps these viruses to attach to the host cells. And most viruses, they specifically infect certain types of cells within one host. And these viruses as well, they have a host range. And this host range is determined by the attachment sites or the cellular factors that allows these viruses to grow. So for instance, in terms of the host range, we have plant viruses that the plants, they have in their cells specific receptors that binds these viruses, but animal cells, they don't have these receptors. And then we have animal viruses that do not infect plants. So that's the specificity in the host range. And also within the host, we have viruses that attack or then that they infect specific cell types. Like for instance, we have viruses that only infects the liver cells, but no other cells in the body. So viruses vary in size, and for the most part, most viruses are smaller than bacteria. So here we have in this cartoon, this bacteria E. coli that measures 3,000 to 1,000 nanometers in their dimensions. We have the largest virus like Ebola virus that measures 970 nanometers that is not as big as E. coli. And then we have other ones like picornavirus, which is the family or the species of polioviruses, and they measure 30 nanometers in their diameter. So since viruses are very tiny in comparison to even the elementary bodies of chlamydia that you can see here, which is a bacteria, and of course super small in comparison to a cell, like in the case of the red blood cells in humans, they cannot be seen with light microscopy. So you have to use a transmission electron microscope or a scanning electron microscope to see these viruses. A complete viral particle is known as virion. And these viruses, they have, again, either DNA or RNA, and they have to have a capsid, which together, the nucleic acid and the capsid, the protection for this nucleic acid is known as nucleocapsid. And this nucleocapsid, if you see the small units that makes the nucleocapsid, this nucleocapsid will be made out of small proteins units that we know as capsomeres. And within these nucleocapsid, you have spikes that are made of proteins. So naked viruses, then they have only DNA or RNA plus the nucleocapsid. And then that nucleocapsid has different shapes. It can be helical, it can be icosahedral, in which you have around 20 facets, and they can be complex. Now, viruses that we consider enveloped, in addition to having this nucleocapsid, they will have a plasma membrane that surrounds the nucleocapsid, or it's outside of the nucleocapsid, more properly saying this. And then within this envelope, the viruses, they have spikes made out of glycoproteins. And the viruses can be classified as resistant or non-resistant to either, depending on whether they can be destroyed or not by this ether. And this is like a chemical classification of the viruses in general. So here we have some examples of how the nucleocapsid looks in naked viruses. So here we have a polyhedral naked virus. So we have only a capsid, which is made out of different proteins that we call capsomeres. And inside this capsid, we have the nucleic acid. And if you see, this is a transmission electron micrograph that shows you this polyhedral naked virus with the spikes protruding from the nucleocapsid, and you can even see these little dots that basically show the capsomeres. Here we have an example of a helical envelope virus. So here the nucleocapsid is helical, and then you have the nucleic acid inside the nucleocapsid, and outside of this nucleocapsid you have this plasma membrane with the spikes, which is basically the plasma membrane from the host cell. So this is the actual transmission electron micrograph of an influenza virus with the spikes, and then it has the capsid inside the plasma membrane, and then it has the nucleic acid. And then here we have an example of a helical naked virus. So here's the capsid, and then here we have the nucleic acid. And this is the transmission electron micrograph of this helical naked virus, which is Ebola virus. So in a scanning electron micrograph, we can see, for instance, a better view, like a three-dimensional view of these capsomeres. So this is a type of virus that's called masked adenovirus, and these are animal viruses. So this is masked adenovirus as seen with a scanning electron micrograph. All of these are individual viral particles with the capsomeres. And then in here, we have this herpes simplex virus that you can see in this transmission electron micrograph the capsomeres, and this is the nucleocapsid, of course, and outside of this nucleocapsid, you can see the plasma membrane of this herpes simplex virus. Also, we have RNA animal viruses. So the previous images were from DNA viruses for animals, and then this one will be for RNA animal viruses. So here we have rhabdovirus that it has like kind of a bullet shape. And then in here we have the nucleocapsid and the RNA. So this is an RNA virus, and this is the virus that causes rabies. And then in here we have this virus that is known as filovirus with the spikes. So this is a naked virus. And then we have complex naked and complex envelope viruses that doesn't have this icosahedral or this helical shape. So in this case, this is a T-even bacteriophage. So this is a virus that infects bacteria, and it has a head. Inside the head, it has the DNA, and then it has this neck that attaches to this sheet, and then it has at the end of the sheet, it has this pin plate, this pin attached to this pin plate, and then from the edges of this pin plate, this virus has tail fibers. So these tail fibers are used by the virus to bind into the receptors of the bacteria, and then with this pin that it has in the base plate, so this will be the base plate, it will inject the DNA. So this that you see in yellow color, this is a transmission electron micrograph. This part that you see in yellow color represents the bacteria. This will be the T-even bacteriophage, how it's attaching into the surface of the bacteria and then injecting its DNA to infect the bacteria. And then we have here an orthopox virus with the nucleic acid here, and then in here we have the outside of these particles of the virus, and this virus, it's a complex envelope virus. So this will be the envelope, and this will be the nucleocapsid. This is a transmission electron micrograph of this orthopox virus. Now, how do we classify viruses? So we classify viruses into families, genus, and species, just similar as to what we do with bacteria. Family names of viruses ends in Viridae, genus names end in virus, and viral species, they have sometimes common names, while they have a generic name for species, and subspecies will be designated by a number. Now, what is a viral species? A viral species is a group of viruses that shares the same genetic information and the same host or ecological niche. So here are some examples of how do we name viruses. So for the family of herpes viridae, remember the ending viridae represents family. So we have a family of viruses that are known as herpes viridae, and within this family there's a genus that is called herpes virus. And we have subspecies of herpes virus that we have the common name. For instance, for human herpes virus, this will be the common name, but the subspecies will be human herpes virus type 1, human herpes virus type 2, and human herpes virus type 3. And you can abbreviate actually the subspecies of different viruses using capital letters of abbreviation of the different names of the species, like human herpes virus will be HHV, all capital, and then with your dash. And for representing type 1, you just put the Roman numeral 1, and then you keep on doing that for the rest. Another example, in this case for a virus of the Retroviridae family, we have the genus, which is lentivirus, and we have these two subtypes. So we have human immunodeficiency virus type 1 or HIV1, and we have human immunodeficiency virus type 2 or HIV2. In order to grow viruses in the lab, you cannot use a petri plate with normal media that we use for growing bacteria or growing fungi. You have to use living cells. And these living cells can be bacterial cells if you want to grow bacteriophages, which are viruses that infect bacteria, or has to be animal cells for animal viruses. Now, when you grow in the lab these bacteriophages, you will form plaques or gaps in the growth of the bacteria on the surface of a petri plate. Now, in the case of animal cells, you have to use special containers that hold cell cultures, or sometimes you can grow the viruses in living animals or in embryonated eggs. And then the cell cultures that we use to grow animal viruses are what we call continuous cell lines. So basically, it will be cell lines that typically are immortal, meaning that they are derived from a cancerous type of cells. So here is the top view of a petri plate with bacteria, E. coli, that it was infected, this bacteria, with the viruses. So all of these surface in solid color, kind of beige, this will be the bacteria. And see all these little dots, these gaps? This will be the bacteria that was destroyed by the virus. When the virus was growing inside the bacteria, it will burst the bacteria, it will kill it, and then you will have these gaps in the surface, in the lawn of the bacteria, and these will be called plaques. Now, for animal viruses, you will use some cells that you previously had treated with enzymes to separate the cells, and then you put them in a special container that has culture medium. And when these cells reaches the bottom of the container, they start growing, and then they form these groups of cells that now can be either transformed cells derived from tumor cells like HeLa cells that are derived from cervical cancer, or it can be cells that are normal, that are not transformed. And then you put your sample of virus in here, and then you will study what is going on if the virus causes some damage to these cells. Now, viruses for diagnosis or sometimes even for developing vaccines can be injected or inoculated into embryonated eggs. So here we have different places where you can inoculate these viruses. So you can do it in this chorioallantoic membrane, or you can do it in the amniotic fluid. This is known as amniotic inoculation, or you can do it in this place, which is called the allantoid. So this will be allantoic inoculation, or you can do it in the yolk sock. So viruses can be identified based on the effects that they cause in the cells that they infect, which are known as cytopathic effects. Cyto for cells, pathos for disease. So what will be the damage that causes in these infected cells? And viruses also can be identified by detecting antibodies against these viruses in the serum of the patient. These are known as titers. And for instance, we can measure the titers of antibodies against hepatitis B, against hepatitis C, etc. And then we can use as well antibodies to identify viruses in what we call neutralization tests, or to clump red blood cells into what we call viral hemagglutination, or we can detect the proteins of certain viruses in what we call Western blot. One rapid way and very accurate sometimes way to detect viruses is by amplifying the DNA or the RNA of the virus into what we call polymerase chain reaction. So you need to have just a little piece of the nucleic acid, and you can make multiple copies with PCR. So here we have an example of cytopathic effect. So this is a fibroblast. The normal view of a fibroblast, which is a cell of connective tissue, seen with the scanning electron micrograph. This is the nucleus, this is the cytoplasm. So when you infect these fibroblasts with viruses, you will clump and basically kill these fibroblasts. So this is what we call cytopathic effect, or CEP, or CPE, sorry. Now, viruses, they don't multiply just as bacteria by binary fission. They multiply by making copies of themselves inside host cells using the machinery of the host cell to replicate. So they use the enzymes like polymerases to make the nucleic acids, or they can use the ribosomes to make proteins. And viruses, they cannot live outside of the cells. And when viruses multiply, they make millions of copies. It's not like one virus multiplying makes two makes four. No, they make millions of copies. And typically the time for making a virus, the generation time is more or less like 4 hours. Some of them, of course, they're much faster than others or slower than others. And then viruses, they can multiply using two cycles, either lytic cycle or lysogenic cycle. So in lytic cycle, the viruses infect the cells and then burst the cells, causing the death of the host cells. And then in lysogenic cycles, the viruses can introduce their nucleic acid into the host DNA, forming a hybrid or a provirus, and then through this, they can either activate certain genes that can cause cancer, or the virus can live in these cells for a long period of time without killing the cell. So in lysogenic cycle, the virus doesn't kill the cell, but it can go into lytic cycle if it opts to do so, and then it will kill the cell. Now in lysogenic cycle, sometimes what happens is that you can have something that's called phage conversion or a specialized transduction. So here are the steps of the lytic cycle. So the virus will attach with, in this case, will be the lytic cycle for a bacteriophage. So the virus will attach into the receptors of the bacteria using stealth fibers, and then it will penetrate by releasing an enzyme that is called lysozyme. The lysozyme is going to break down the wall of the bacteria, and then the tail sheet will contract, forcing the tail into the cell wall, and then the DNA will be injected into the host cell. And then once inside this DNA of in the host cell, the virus is going to start forcing the cells to produce the nucleic acid of the virus. So basically, viruses are parasites, and then also will force the cell to make proteins. Once the virus has DNA pieces and proteins, or well, not pieces of DNA, it will have copies of DNA and proteins. It will assemble these into what we call the maturation stage. And then once the cells have fully complete virions, then the cells are going to burst. So the release is going to be by breaking down the wall and the cells will die. The viruses, when they're doing that, they don't show particles in the medium where you're growing the bacteria. So this is known as eclipse period, in which when you introduce, let's say, at time 0, so this is like a graph showing you the growth of viruses within host cells. So in the y-axis, you have the number of virions. In the x-axis, you count the number of days that takes for detect the viruses in the medium where you're growing them. So if you inject, let's say, 100 viral particles on day 0, then let's say by day 3, the viruses disappear. This is known as eclipse period. You don't take viruses. Why? Because the viruses infected the cells and they are being assembled inside the cell, so you cannot detect the viruses outside the cell. Now, when the viruses start growing and getting assembled in the maturation process, they will burst the cells and then they can be detected because they will be released from the host cell, and this is what you call an acute infection. Now, eventually, if the viruses run out of cells to infect, the viruses will decline in numbers until they reach day 0 because they don't have other cells to infect. This is just to show you the steps of the lytic cycle of what we call a T-even bacteriophage. So in here, we have the attachment of this virus into the cell wall of this bacteria. And then if you magnify, you will see how this virus uses the tail fibers to attach into a cell wall and then contracts the sheet, introduce the pin, and then the virus is going to penetrate. So it's going to inject the DNA into the host cell. And then in biosynthesis, the virus is going to start making pieces of themselves, like it's going to start making the head, it's going to start making the sheet, the tail fibers, the base plate, and it's going to make copies of the DNA. So it's going to assemble in the maturation step. So as you can see here, it will move from this part up to a complete virion, and then it will release by bursting the cell. So this is the lytic cycle. Now in the lysogenic cycle, which is in this part, the virus which have injected the DNA into the host cell, it will integrate the DNA into the host cell, making what we call, in this case, a prophage, but it can be called a provirus. And then the virus will basically make copies of themselves in this lysogenic cycle as the bacteria is dividing by binary fission. So as you can see, this pink part of this DNA represents the virus DNA, and in this case, the purple will represent the bacterial DNA. So when the bacteria makes a copy of themselves, also copies the virus DNA. So you end up with bacteria with viral particles in a hybrid mode. And then the virus, if it opts to go into the lytic cycle, it can separate its DNA from the host cell, and then it will enter and form new particles. So why does the bacteriophage will alternate between cycles? So let's say that the viral particle detects that there is low nutrients within this medium that the bacteria is growing. So in this case, we'll undergo the lysogenic cycle. It will prevent the bacteria from bursting. But if there is a lot of nutrients available, in this case, it is convenient for this phage virus to multiply and burst the cells, and then will release into the environment to infect new cells. And then sometimes what happens with these bacteriophages is what we call transduction. So in this case, the phage will infect this donor bacterial cell, and then the phage DNA will make proteins, and the chromosome of the bacteria is broken into pieces. When the bacterial chromosome is broken into pieces during maturation stage, the DNA of the bacteria can go into the head of these virions, and then the phage DNA will go into these particles. So now, when a bacteria, a new bacteria, which is going to be recipient, receive this bacteria that, this virus that carries the bacterial DNA and infects the new cell, you will have a donor bacterial DNA. So let's say that this was a gene that had basically resistant to antibiotics. So in this case, this capsid or this head of this bacteriophage or this virion, now it will introduce into this bacteria that it wasn't resistant to antibiotics, it will introduce a piece of DNA that it will make the bacteria to be resistant to antibiotics. And then as the bacteria is duplicating in binary fission, it will make a clone of cells that now is going to be resistant to antibiotics. And similar to here, but this is called specialized transduction. So in this case is when you are in the lysogenic cycle, you have a prophage, and then when this prophage separates this viral DNA from this original bacteria, which was what we call the donor, now this prophage can carry this galactose gene from this galactose positive bacteria. These galactose positive bacteria basically can grow in a medium that is rich in galactose. And then when this DNA from the virus excises or separates from this bacterial DNA, it will carry this galactose gene, and then this mature virion that now is going to infect a recipient cell, when this recipient cell integrates the DNA from the virus, now this bacteria that previously was galactose negative, it couldn't metabolize galactose. But now with this galactose gene that it was carried by this virus DNA, now the bacteria becomes galactose positive and can metabolize galactose. So this is what we call specialized transduction. Now, the viruses in animals also have multiplication steps. So these are the following. So they are similar to the bacteriophage steps. So the viruses, they have to attach into a receptor, but now in the cell membrane. And then the virus will penetrate the animal cell either by endocytosis or by fusion. And then once inside, the virus will uncoat. It will basically release the capsomeres and release the nucleocapsid and the host enzymes. This is known as uncoating. And then once the virus has uncoated, it's going to make copies of the nucleic acid and the proteins. And it can be early proteins and can be late proteins. And these early proteins allows the biosynthesis of more viral particles. And then the virus will mature. So you will assemble the pieces, and then you will have virions, and then you will release these animal viruses by budding or rupture of the cells. Budding if it's an envelope virus, and rupture if it's a naked virus. So this is the transmission electron micrograph enhanced with color to show you the entry by endocytosis or by fusion. So in this case, you have this virus, which is a retrovirus, binding into this plasma membrane of the host cell, and then the host cell will encase the virus, forming a vesicle. This is known as endocytosis. Now, in the case of this envelope virus, which is a herpes virus, the virus will leave the envelope fused with the plasma membrane of the host cell, and then it will just introduce the nucleocapsid with its DNA. And this is just to show you the release of the virus by budding. So here we have viral entry in this transmission electron micrograph, and in here we have the exit of viruses. So in here we have the viral capsid, and then you have the host plasma membrane with the viral proteins inserted into it, and then the virus is going to bud. So you have a protrusion of the plasma membrane and then pinching off of this and released by buddings, which you can see here with this lentivirus in this transmission electron micrograph. So this will be the different steps. So this will be corresponding to this last step. And just to show you again, the viral entry with mediated endocytosis for naked virus. So viral particles, in this case with this transmission electron micrograph, will be attaching to the host cell plasma membrane, and then you will introduce the plasma membrane with a particle forming an endocytosed viral particle. And then for fusion, you have the viral envelope here of herpes virus that contacts this plasma membrane of the host cell, and then the plasma membrane of the virus fuses and is left behind, while the nucleocapsid and the nucleic acid enters and then will be released into the cytoplasm. And this is just to show you the cartoon of the cycle or the steps of the animal virus attachment. So the viral particles again attaches into the plasma membrane. They enter and they are encoded. So they release the capsomeres of the capsid and then the nucleic acid, which in this case is viral DNA, and the particles will enter into the nucleus so that they can be making copies of the proteins, the early proteins, which is known as early phase proteins. So this is the messenger RNA that exits into the cytoplasm of the host cell. And here in the cytoplasm of the host cell, you will make the capsid proteins, and you will start making as well, while this happens, copies of the DNA, and then the capsid proteins will re-enter into the nucleus so that you can mature the virion particles inside the nucleus, and then you will exit this. So this is the typical example of papovavirus. Other viruses, they perform, in the case of DNA viruses, most of them, they do so their steps in the nucleus, but some of them make them here in the cytoplasm. For RNA viruses, it depends on what type of RNA virus you have. If you have plus stranded or negative stranded, plus will be an RNA that runs from 5' to 2 from 3' from 5' to 3', which are the ends of the nucleic acid, and negative strand will be the opposite. Some viruses, in the case of the plus strand, single-stranded RNA, like picornaviruses, they directly form proteins because they can be taken as messenger RNA. And then they can make the proteins, of course, in the cytoplasm where you have the ribosomes, and they will make copies of the also the nucleic acid within the cytoplasm, and then they will mature, and then they will be released. So basically, for most of the viruses, the RNA viruses, the maturation and the entry is the same, but there is a difference in where the viruses are made and how they are made. So single-stranded RNA or negative antisense, like in the case of rhabdovirus, which is the virus that causes rabies, in this case, you will have messenger RNA plus strand first transcribed from the negative viral genome, and then you will do that in the cytoplasm, of course, and the viral genome will be made before the proteins can be synthesized. And then you will make your capsid proteins, you will assemble the virus, and then you will release it. In the case of double-stranded RNA, the double-stranded RNA can be positive and negative strand, like in the case of reoviruses, and then you can form the messenger RNA inside the capsid and release into the cytoplasm of the host. And then you will mature these particles, and then you will release them. So for messenger RNA then for RNA viruses, you can have negative strand or antisense, positive strand or sense, and you can have single-stranded or double-stranded. And if it's double-stranded, it has to be plus sense and antisense. Now we have special types of viruses, like in the case of retroviruses. Retroviruses, they have the opposite of what we know as forward transcription. So regularly we make out of DNA RNA copies, but in reverse transcription we will make a copy of DNA out of RNA. So we have viruses like in the case of HIV virus that has an enzyme that is called reverse transcriptase that allows them to make a copy of DNA out of an RNA virus. So in this case, the virus will bind into the host cell, fuses its envelope, and then will encode, and it will release the viral RNA, and it will release the reverse transcriptase. Now, the reverse transcriptase is going to make viral DNA, and this viral DNA, it will go into the host cell nucleus, and it will integrate with an enzyme that is called integrase the viral DNA into the host cell DNA, making what we call a provirus. So this is a hybrid. Part of this DNA is going to be from the virus, and part of the DNA is going to be from the host cell. Now, this provirus can live for as long inside the nucleus of the cell and inside the cell, of course, for as long as the cell will live. And this virus then can remain latent or in the cell for long periods of time, infecting the cell. Now, the provirus can make copies of themselves by making RNA within the cell, and then will make viral proteins, and the virus is going to be assembled within the cytoplasm, making a new viral particle. And then this viral particle is going to be budding, taking plasma membrane, and then it will take also the spikes made of glycoproteins that allows them to infect a new host cell. So this is the life cycle of a retrovirus. So as a summary, There are some differences, minor differences between the multiplication of bacterial viruses and animal viruses. Bacterial viruses are known as bacteriophages. Why? Because they eat bacteria. So basically, bacteria for bacteriophage for eating bacteria. So in this, the attachment, so bacteriophages, they use tail fibers that attach to the cell wall. In the case of animal viruses, they attach into receptors in the plasma membrane, which are proteins or glycoproteins. The entry for bacteriophages, you will inject the DNA. And for animal viruses, you can have entry through endocytosis or viral fusion. Encoding for bacteriophages is not necessary, but it is necessary for animal viruses. So they release their capsomer proteins. And then the multiplication happens in the cytoplasm for bacteriophages. And in animal viruses, if, for the most part, if it's a DNA virus, happens in the nucleus, while if it's an RNA virus, happens in the cytoplasm. Bacteriophages can lead to lysogeny, and in the case of animal viruses, will lead to latency. And these will lead to slow viral infections and sometimes to cancer. And bacteriophages, they can lyse the host cell, while in the case of animal virus, you can have budding out of the viruses or rupture if it's a naked virus. Now, in this part of the presentation, we're going to see how viruses can cause cancer. We're going to differentiate different stages of viral infection. So we're going to see what is a persistent viral infection, what is a latent viral infection. We're going to differentiate viruses, viroids, and prions, and we're going to see what are the modes of infection of prions. We're going to list some viruses that infect plants and some viruses that infect humans. 10% of cancers are caused by infections of viruses. Viruses can activate normal genes that we have in our DNA that are known as oncogenes. These oncogenes, when they are activated, are known as proto-oncogenes, and they can transform the normal cells into cancer cells. And the genetic material of oncogenic viruses can become integrated into the host cells, forming what we call proviruses. And these proviruses, if they integrate in the DNA of the host cell, that it is an oncogene, as these viruses are within this area next to the oncogenes, it can make the oncogenes to be activated, transforming the cells. What are transformed cells? Cells that are constantly dividing without any purpose. So they have an increased mitotic rate, so they form a tumor. And cells regularly, when they are growing, they're supposed to have physical contact and stop growing. This is known as contact inhibition. But in the case of transformed cells, these cells are growing and growing and growing. They're making copies and copies and copies. They contact one another and they don't stop growing. This is known as lost contact inhibition. So that's why we end up with tumors. And then in the case of tumor cells that are induced by viruses, these viruses, they sometimes have antigens that are displayed in the plasma membrane, and this will be called virus tumor specific transplantation antigens, or TSTA. And some viruses, they can display the antigens, which are proteins basically, in the nucleus. So these are known as T antigens. We have different families of oncogenic viruses. So we have families of DNA viruses and families of RNA viruses. So DNA oncogenic viruses Oncogenic means that are capable of forming tumors. So we have adenoviruses, we have herpes viridae family members like Epstein-Barr virus, we have human herpes virus type A that causes Kaposi sarcoma, which is a cancer in the skin and other tissues. We have members of the Poxviridae family. We have members of the Papoviridae family, like human papillomavirus 16 and 18 that causes cervical cancer. Also, they can cause cancer in the anus or also in the larynx. And then we have hepatnaviridae members like hepatitis B virus that causes liver cancer. We have RNA oncogenic viruses like human immunodeficiency virus. We have human T leukemia virus or lymphotrophic virus like human T leukemia virus type 1, human T leukemia virus type 2. We have RNA viruses that causes also cancer in the liver like hepatitis C virus, which is part of the Flaviviridae family. We have also oncogenic viruses that affects domestic animals like cats, dogs, chicken, sarcoma viruses. We have feline leukemia virus that causes leukemia in cats. And here is just to show you an example of an oncogenic virus. So here we have a tumor within the neck. This is what we call Burkitt's lymphoma, that it is caused by an DNA oncogenic virus. Now, viral infections can be acute, in which the infection lasts for a certain period of time and then it's basically not happening once the virus is killed by the host cells or the immune system of the host. But we can have viral infections that persist, that we know as latent. Now, in this case, we have the virus living in the host cells for as long as the host will live. And these viral infections, latent or persistent, will be in some cases asymptomatic, like in the case of when someone initially had cold sores, but this cold sores doesn't appear anymore. But when the person becomes immunocompromised, the cold sores return, or also in the case of shingles. Now, in some cases, some of these latent or persistent viral infections can be activated or reactivated in such a strong manner that it can kill the host, like in the case of measles. So measles virus can cause subacute sclerosing panencephalitis, that is a fatal infection that affects the brain tissue. So here is the difference between an acute infection and a persistent or latent infection. So in acute infection, the infection lasts for days, while in persistent and latent infections, the infections last for months up to years. And then you have remissions and exacerbations of the signs and symptoms. So here's an example of herpes virus infection reactivation. So when a person was young, in this case was infected by this virus that causes varicella during childhood, the infection can go away, but it can go into the nerve cells of the dorsal root. In this case will be for the intercostal nerves. And then this virus will be remaining latent for years. And when the person is an adult and it gets immunocompromised, this virus will activate again, will make copies of themselves, and will move through this nerve, the intercostal nerve, and will cause herpes zosters or shingles, causing infection in the area where the nerve is. And this causes a lot of pain. Similarly, for cold sores. So these cold sores will cause these vesicles in the commissure of the lips, and then with time, this virus will go into the ganglion of the trigeminal nerve, and then it will reactivate itself when the person is immunocompromised, and then the person ends up with vesicles in the perioral area. Now, prions are proteinaceous infectious particles. So basically are pathogens made out of proteins. This is fairly new to us. We never knew about it until a couple of decades ago, in which we found persons that were infected with what we call the chip scrapie, Creutzfeldt-Jakob disease, German Streuhold Schenker syndrome, fatal insomnia, and mad cow disease. So all of them, they share in common that infects the brain and causes hole in the brain that makes the brain tissue look like if it was a sponge. So generically speaking, these infections are known as spongiform encephalopathies. Now, the way that the person can get infected with these proteins is by ingesting contaminated meat of cattle or even sheep, and by having transplants from persons who were having these infections, or by the person having a surgery with contaminated surgical instrument with these prions. Now, prions infects the brain tissue because in the brain we have these normal cellular prion proteins that are called PRPC in the cell surface of neurons. Now, these PRPCs can bind to these prions, and then they will transform into scrapie proteins that we know as PRPSC. And this will precipitate proteins within the brain cells forming plaques, and then they will cause these holes in the brain tissue. Prevention for having these prions contaminating surgical instruments is by autoclaving longer period of time these surgical instruments that were contaminated for more than 15 minutes and at a higher temperature, at a temperature of 134 degrees Celsius. But these instruments previously also had to be treated with sodium hydroxide to denature the proteins that causes spongiform encephalopathies. So this is the cycle of infection of prions. So here we have these normal proteins in the brain tissue. So this represents basically a neuron. So we have PRPC, which are the receptors that are going to be bound by these prions. So prions will bind then into these receptors, and they will convert these normal proteins into scrapie. So these scrapie are going to be endocytosed by the cells, and it's going to precipitate forming abnormal proteins. And they will cause cell death within the neurons, and then you'll have the following. So this is a light microscopy view of these holes that are caused by cell death in the neurons. So this is a light microscopy view postmortem of someone who was affected by an spongiform encephalopathy. And this is a transmission electron micrograph showing you the PSPRC, the precipitated proteins in the neurons. Now, plant viruses are viruses that enters through the wounds or via insects into plants. And plant viruses are made out of a nucleic acid, just like as for animal viruses. And we have also viroids that are short sequences of RNA that can infect plants as well, but instead of having a long polymer of nucleotide bases of RNA, we only have a short base. So here we have an example of a long linear plant virus, and here we have these little viroids, short sequences. So here we have a table showing you the different families of viruses that can affect plants. So here we have a double-stranded DNA virus, non-enveloped, that is called cauliflower mosaic virus. So this is the morphology, is icosahedral, and it is transmitted by this insect, aphids. And then we can have also single-stranded RNA viruses, plus strand, negative strand, and double-stranded RNA. So we can have basically different types of viruses. And in this case, for single-stranded RNA negative strand envelope, we have Rhabdoviridae family members like potato yellow dwarf virus that is basically out of the family member of rhabdoviruses that causes rabies in us. And here we have the mode of transmission. It is transmitted through leafhoppers or aphids, while in this case for virus, it enters into the plants by wounds. Now let's look at some virus families that affect humans. And we're going to understand what are some of the diseases that are caused by DNA viruses and others like RNA viruses. So for DNA viruses that are non-enveloped, so for the most part, viruses that are DNA are double-stranded with a few exceptions. So in the case of single-stranded DNA, so will be parvovirus family. So these will be single-stranded DNA, no envelope viruses. They are small, between 18 to 25 nanometers in diameter. An example would be human parvovirus B19 that causes a disease that is called Fifth disease, or it can cause anemia in immunocompromised patients. Double-stranded DNA, we have also non-enveloped or naked viruses like adenovirus that measure between 70 to 90 nanometers. We have an example, an important general like mass adenovirus, that it can cause respiratory infections in humans and it can cause tumors in animals. Now for Papoviridae family, it measures between 40 to 57 nanometers. This will be important for us because it has this papilloma virus that causes human warts, and this will be this family that has species like human papillomavirus 16 and 18 that can cause cervical and anal cancer. And then some of the other papillomavirus members, which are basically hundreds of members, they don't, well, it's a little bit over 100, not hundreds, but they cause warts. And sometimes they can cause warts in the genital area like condylomas. And then another virus that we have in here is polyomavirus. Double-stranded DNA envelope viruses. So these are a little bit larger than the previous ones, like pox virus. They measure between 200 to 350 nanometers. And we have important members of the orthopox virus genera like vaccinia virus and smallpox or molluscu pox virus. So we have smallpox that causes smallpox, variola, or molluscum contagiosum, which is basically a lesion in the skin that is like a raised area that has like a little dimple in the middle. And then cowpox virus, it's another member of this family. Herpes virus are medium-sized viruses. They measure between 150 to 200 nanometers. They are envelope viruses, and we have different members. We have human herpes virus from 1 through 8. These are the different names. And we have examples of diseases caused by human herpes viruses. So, for instance, we have chickenpox, shingles, fever blisters, mononucleosis, infection mononucleosis for cytomegalovirus, Burkitt lymphoma, we have cold sores, and we have Kaposi sarcoma for human herpes virus type 8. And then we have roseola caused by human herpes virus type 6 and 7. Hepataviridae is a small virus, measures 42 nanometers. It has hepatitis B virus as an important genera. And then we have this virus that it has this reverse transcriptase, hepatitis B virus, and causes hepatitis B and cancer in the liver. So here are just some images to show you smallpox lesions. This virus was eradicated in the 70s, and this was eradicated because of the efforts of vaccination. But this virus used to cause these vesicles in the skin of the person, and inside these vesicles, you have not only pus, but also you have the viral particles. So anytime that we have an infection in the skin that is caused by viruses like herpes virus, cold sores, or varicella, it is advised that persons don't scratch the lesions because when they rupture these vesicles, inside the vesicles you have the virus and you spread the virus. This is roseola virus causing this rash in the skin of the person by this virus, of course, and this is caused by roseola virus. Single-stranded positive strand RNA viruses envelope are the following. So we have togaviridae, flavivirus, and coronaviridae that now is causing this pandemic that hopefully it will end up soon. So coronaviruses can cause upper respiratory tract infection. It can cause as well infections of the digestive tract. And we have different subspecies that you know because we have mutations in these RNA viruses. These RNA viruses, as they're making copies of themselves with this RNA polymerase enzyme, this RNA polymerase enzyme basically introduces mutations in the DNA, and that's why, unfortunately, we don't have a successful vaccine that can cover all the subspecies of this virus. However, of course, it's important to get vaccinated because some of these vaccines that were developed against the most common types of subspecies that we have. And then we have other members like alpha virus, like alpha rubivirus, that it is causing rubella. And then we have alpha viruses that are transmitted by arthropods that causes Eastern equine encephalitis, Western equine encephalitis, and rubella virus that is transmitted by respiratory route. Now, in the case of flaviviruses, we have an important member like hepatitis C virus that causes hepatitis C and cancer of the liver, and others like in the case of flaviviruses that are transmitted by arthropods causes yellow fever, dengue, St. Louis, and West Nile encephalitis. So this is rubella, causes a rash in the skin. And then 4 negative strand single-stranded RNA viruses. So we have rhabdoviruses that has a bullet shape. We have flaviviruses that has like a rod shape. We have paramyxoviridae, which are basically viruses that are complex. Deltaviridae that are defective viruses that are small. And for rhabdoviridae, they can cause rabies in numerous animals. So this is a zoonotic disease that can infect humans and also can infect raccoons, bats, skunks, etc., wild animals. So this one is transmitted by bites, and this is a virus that infects rhabdoviruses, infects the nervous system. In the case of filovirus, which one of the members will be Ebola, it can infect the endothelial cells of the capillaries and can cause hemorrhages. Paramyxoviridae, like members like parainfluenza virus, mumps, infects humans in the digestive system for mumps. It infects the parotid glands. And we have animal viruses that infect chickens, like Newcastle disease in chickens. Delta viridae has hepatitis D virus that is defective. This is a virus that is naked and it requires the envelope from hepatitis B virus to infect the liver. So it co-infects the liver together with hepatitis D. Otherwise, itself, it cannot cause infections. So this is the example of a case of mumps infecting the parotid glands. Mumps virus. Single-stranded virus RNA that is plus strand, non-envelope. We have two families. We have Picornaviridae family and Caliciviridae family. In Picornaviridae family, you have enteroviruses, rhinoviruses, and then we have hepatitis A virus. Rhinovirus is the agent that causes common cold, and enteroviruses infects the enterocytes or the digestive system, causing diarrheas. And for an important member of the Picornaviridae family, we have poliovirus that causes polio, that infects the motor neurons, leaving the person with problems for moving their muscles. And sometimes it affects so much the nerves that the person end up with a limb that it is basically atrophic. And then we have echoviruses, we have hand and foot mouth viruses within this family, and we have around 100 rhinoviruses that causes common colds. And then for Caliciviridae family, we have hepatitis E and noroviruses. They are both involved in cases of gastroenteritis, but in the case of course, hepatitis E infects the liver, causing hepatitis. Negative strand, multiple stranded RNA viruses. So we have Orthomyxoviridae family, Bunjaviridae, and Arenaviridae. Arenaviridae is kind of a strange virus because it has within its DNA pieces that are plus strand, pieces that are negative strand. Although it's strange, this virus has an important shape because it looks like granules. So that's why it's called Arenaviridae. It looks like sand granules. And then we have, in the case of Orthomyxoviridae, we have influenza virus type A, type B, and type C that causes influenza. And they have in their envelope spikes that can agglutinate red blood cells. For Bunjaviridae, we have California encephalitis and hantavirus that causes hemorrhagic fever. And this hantavirus has been known that is transmitted by rodents in the feces or in the pee. So whenever someone ingests this virus, because the person may have perhaps have drink from a can that it was contaminated with feces or pee of the virus, then the person will ingest this virus and it will develop pulmonary syndrome. Now, that's why it's important then to every canned food that you will be ingesting from to clean it really well. And then in the case of these arenaviridae, we have Venezuelan hemorrhagic fever, Lassa fever, and also lymphocytic choriomeningitis. So basically, these will be infections of the brain tissue. Retroviridae and double-stranded RNA. So most of the RNA viruses are single-stranded, with the exception of reoviruses, which are either well, Reoviridae family members like reoviruses or rotaviruses. These are double-stranded RNAs that are naked. But most of the RNA viruses are single-stranded RNA. And in the case of this single-stranded RNA that produces DNA, which is a retrovirus, this will be HIV virus, which is part of the lentivirus genera. So this will cause HIV, and we have oncoviruses from this family that causes leukemia and tumors in animals. And then for reoviruses and rotaviruses, they can cause mild respiratory infections that are transmitted by droplets. Now, this is a comparison of hepatitis viruses. So we have, of course, viruses from different types. We have RNAs and RNA viruses and DNA viruses. Now, hepatitis A and E are transmitted by fecal oral route, meaning that someone will ingest these viruses. So that's why here the mode of transmission is ingestion. These other viruses, B, C, and D, are transmitted parentally, basically through the skin, or by sexual contact in the case of hepatitis B and hepatitis C. And the incubation period, which is the time that it takes for the virus to make the host to have signs and symptoms. Well, hepatitis A and E, they have similar incubation period between 2 to 6 weeks. These other ones have a better period, it's a little bit longer. And some of them, they can cause chronicity, chronic infections like hepatitis B and C. And they can cause basically cancer for hepatitis B and C of the liver. And then vaccination available, we only have vaccination for hepatitis B and hepatitis A. But for the rest, C, D, and E, we don't have vaccination. And hepatitis B is protective vaccination for hepatitis D. Treatment will be none for hepatitis D and E, immunoglobulin for hepatitis D, which is A, which is antibodies, interferons for hepatitis B and C, and ribavirin, which is basically antiviral for hepatitis C. And also we have antivirals for hepatitis B, but these are called nucleoside analogs. And this is just to show you what happens with an infection, chronic infection with hepatitis C. So here's a healthy liver. This will be the right lobe, the left lobe. The surface is pretty smooth. The color is kind of reddish brown or dark red just because we have a lot of red blood cells. But this is what happens with time with someone with hepatitis C. So you can develop tumors and, of course, shrinkage of the liver and dysfunction of the liver and ultimately death. And then for comparison of arboviral encephalitis, so what we're showing here is that certain virus affects, no matter what, if they're of different type, they have tropism. Basically, they infect certain tissues. So we saw in the case of the previous slide, the viruses that infects the liver. Now, viruses that infect the brain. So we have different types, and most of them, they are transmitted by this mosquito, which is Culex mosquito. So the mosquito can inject the virus into the host as it's taking a blood meal from the host. And these viruses, they replicate actually within these arthropods, within these viruses, either in the salivary gland or in the gut. And then most of these viruses then are transmitted by the Culex mosquito, but other ones, they can be transmitted by Aedes species or by Culicera. So depending on the area of United States that it is affecting, you can call it Western equine because you have basically not only the transmission through these arthropods, but you can have also a reservoir, which is an animal or a host that can keep the virus. So in this case, for Western equine encephalitis, we have birds and horses. Equine basically will be because the majority of the reservoir will be horses. So Western equine encephalitis, or WEE, it is a virus of the family of the toga viruses, affects most Western states. Eastern equine encephalitis affects mostly the eastern states, and then St. Louis encephalitis, entire United States. California encephalitis not only affects California but all other states uh, of the east, and also here within the Southwest. And then West Nile encephalitis affects the entire United States. And then mortality rates are the following. So basically, how likely are to produce infections that kills the host. So 5% for Western equine, greater than 30% for Eastern equine, 20% for St. Louis encephalitis, 1% of those hospitalized for California encephalitis, and West Nile encephalitis between 4 to 18. So the most dangerous one then will be the Eastern equine encephalitis. Now, in the case of some viruses, they can undergo what we call antigenic shift, in which you will have changes in the genome of the virus that can lead to changes in the receptors that they have that helps the viruses to bind, and these will be antigens. So we have this influenza virus, which is part of the Orthomyxoviridae family, that can infect humans, but also can infect the avian species and also the swine species. Now, a person can be co-infected with avian viruses or swine viruses. And then you have here the representation of human virus gene pool, avian virus gene pool, and swine virus gene pool. Through close contact with these species, you can have changes. This is a virus that is RNA that has segments. It's segmented RNA. You can have up to 7 segments, and then you can have reassortment of these segments into what we call triple reassortment, sorry. So this is avian gene pool reassorting with the human H3N2 and the North American swine. And all of these antigens in the surface of this virus can change, and then they can produce new variants that causes pandemics. And that's why every flu season we have to see which of these variants is the most prevalent and which one is the most likely to cause a pandemic, and then we recommend people to vaccinate to prevent these from happening, from these pandemics from happening. Lastly, the highlight of this chapter is that synthesis of the viral material and the proteins is dependent on the host cell. So viruses infect a specific host because they have specific proteins that can bind to specific host cells, and then the viruses can replicate inside this host cell. So this is all for this chapter. I will see you in the next. Bye-bye.